Enzyme specificity is often described as a lock and key model by virtually all high school biology textbooks around the world. However, there is a problem with this example of lock and key to explain how enzymes work. First, let's understand how enzymes work. Enzymes enable substrates to attach to them, thereafter causing the substrates to react or be broken down chemically. In short, enzymes boost the chemical breakdown of substrates. In this lock and key example, the enzyme is supposed to be the lock and the substrate is supposed to be the key. However, because the enzyme is responsible for action on the substrate, or because the enzyme boosts the breakdown of the substrate, this model of lock and key is limited, if not plain wrong. Why? In practice, a key is responsible for action on a lock. A key opens a lock, not the other way around. A key basically breaks down a lock. Now, as we have mentioned, an enzyme catalyzes or enables the breakdown of a substrate. An enzyme works on a substrate, not the opposite. In a perfect example, the enzyme function should be like the function of a key, not like the one of a lock. Therefore, the enzyme cannot and should not be given the example of a lock. In comparison, it can only be a key. But if you use the example of the enzyme as the key, it brings the problem of being able to match its role in terms of shape alignment, and you now see why this example used in books all over the world is wrong. Most critically too, the most important trait of a catalyst is that it remains unaffected by the reaction it catalyzes. Here, a padlock example fails flat because a padlock is changed by the action of a key. A better example or analogy would be to imagine an enzyme as a socket and the substrate as a plug. You can call this a socket and plug model. A specific socket can only fit a specific type of plug. For example, a 2-hole socket can only fit a 2-pin plug. This is comparable to how a specific enzyme can only fit a specific substrate. Also, a socket enables electric current to flow into the plug the same way an enzyme enables chemical interaction within a substrate. Essentially, a socket unlocks a plug's function the same way an enzyme boosts the function of substrates. And, as we have mentioned, in addition to specificity, this is the second critical basic concept we need to find in any model used to explain the workings of enzymes and substrates. Finally, and most importantly, a socket can be connected to many similar plugs, one after another, and remain unchanged from these interactions. An enzyme must remain unchanged from a reaction it catalyzes. This is a core character of a catalyst, as all enzymes are or should be.